Hello everyone, my name is Alexander Krämer. I'm a biotech equity research analyst here at ABG Sundal Collier. Today I have the pleasure to host David Westberg. He is the CEO of the Swedish biotech Nanexa. They are based in Uppsala, and today we will have a fireside chat about Nanexa and about the prospects in 2024.
Okay, thank you for hosting us.
Thank you, David, for coming and welcome. First of all, I would like to ask you if you could please provide a short recap of Nanexa for those who don't know the company yet.
Okay, absolutely, I'll do that. So Nanexa is a company that evolved from the University of Uppsala, actually, basically, and that was many years ago. But since 8 or so years, we have been working on a drug delivery technology that we call PharmaShell. That is a technology for long-acting injectables, where we encapsulate small particles of APIs. That could be small molecules, peptides, monoclonals. We can actually, we have now provided evidence that we can do monoclonals as well. So, but what we then do is we encapsulate that with an extremely thin layer of an oxide, inorganic oxide, that slowly dissolves when it comes in contact with water, and then it releases what's inside of it. And by doing that, we can actually design products that have a weekly release or a monthly release or a 3 months release quite easily. So that is Nanexa, and we're about 20 people working in the team, and about another 10 consultants that are more or less on a need to on a basis of when it's needed, so to say.
Mm-hmm. Okay, nice. And let's start with your first, or let's say your lead product at the moment. It's the Next-22 project.
Yes.
There has been quite a lot of interest, of course, I mean, the whole GLP-1 space. And there soon you're going to, or like you're in the preparations of running a phase 1. And I would like to hear from you, like you are collaborating with a German CRO there, and how's your feedback from the CRO? Like, are they ready to start? Like, how's the progress in that project?
Yeah, it is good progress. Profil is a very good CRO for this type of study, specifically in the GLP-1 area. And we have selected them way back just because of that, and we're now in the phase of we have submitted the clinical trial application just before Christmas, and we're expecting to get an approval from the German authorities either in March, or it could potentially be delayed until April. But the CRO Profil is really ready to get started as soon as we get approval from the German authority. We have also the clinical trial material, all that has been released, packed in boxes to be ready to send, so to say. So everything is in place to get started. We know that they have started to look in their database of patients to who could be suitable for this study. Mm-hmm. So we expect to get a good and a quick start after the approval.
Mm-hmm. That sounds very promising. And now shifting gears a little bit, my next question, I mean, you kind of mentioned already a bit about your PharmaShell technology. And one question which I have here around is when you're like playing in the GLP-1 space, one important question that will be about the scale up of the manufacturing and the scale up of the technology. As you know, I mean, for example, Novo Nordisk, they have at the moment supply constraints and really problems of ramping up the production to meet the huge demand. So that's like the first question, like how is it adapted, your technology, to like scale up? And the second part of the same question is about how is your technology compatible with combination preparations? For example, Novo Nordisk, they have CagriSema, which is a combination preparation, if this is something that your technology is compatible with.
Okay, a lot of questions in one question, but I'll try to take them one by one. When it comes to upscaling of our process, we have since 2020 started working with a huge company in the US. The name of it is Applied Materials. They are the world's largest manufacturing of equipment for manufacturing using the ALD technology. So they are huge in the semiconductor industry, for instance. And And they have, and actually, I actually visited them in India last week. They have tools now in different scales. Normally, we work in our lab in about 50-gram scale. We also have a tool for about 200-gram scale, so that is really, really small. But their largest at this moment is a 100-liter reactor, and on the drawing board is a 250-liter reactor. And that will be sufficient for the batch size, at least at the launch of a GLP-1 product. Potentially one would go even further up in size. But the technology as such and the process is easily upscaled, I would say, and very much compared with other unit operations in the pharma industry. This is a quite straightforward technology to scale up. So in place now, there are a number of scales, a number of batch size, I should say, on the equipment, and they have shown upscaling tests with other substances from the scale that we are now up to at least a 10-liter scale, and they will provide more evidence in a 25-liter and so on as it goes. The plan is also that we will have those equipments installed at our facility in Uppsala, so that we can easily do clinical trial material for larger clinical trials as well, and pivotal stability studies and so on. Now, when it comes to Novo Nordisk and their constraints when it comes to manufacturing today, as I believe it is the fill finish part of the production that is limited, and they are of course investing a lot in different sites for doing that. Referring back to our technology in a long acting, of course we will decrease the number of units by about 4 compared to their Ozempic or Wegovy product. So that would be easier, one can say, and it will be about the same type of technology for a fill finish product. So I think in that sense, yes, we will be able to scale it. Absolutely, our process and the fill finish will probably help them in one way of lowering the units actually that needs to be filled. Then the third question I think was if our technology is compatible also to their GLP-1 GIP products that they are developing, and there are actually many programs, or at least a few programs in phase 2 or so.
The CagriSema, for example.
Yeah, CagriSema, for instance. And absolutely, it is also, we have been able to coat peptides, we've been able to coat antibodies, so definitely there shouldn't be a problem to do a 1-month product out of those combination molecules. So we don't see a limitation there, and if Novo Nordisk choose to continue with us, they of course will not use our product just solely for one product. They will use the technology as a platform technology.
Talking about Novo Nordisk, of course, I mean, all eyes are on a Novo Nordisk collaboration at the moment. Do you plan in 2024 to share some updates regarding your Novo Nordisk collaboration?
I would like to share any updates any day, but of course we have constraints there that comes from their agreement with them, and but let me phrase it this way: 2023 has been a very good year for our collaboration. We're continuing into 2024, and I anticipate that we will reach a few of the very, very, very interesting milestones during this year. How far that comes is impossible for me to say at the moment, or at least I cannot say it.
Okay. Besides your own project and the Novo collaboration, you also have a couple of partner projects. Could you provide an overview of the partner projects that you are running at the moment?
Yes, absolutely, I'll do that. And it is interesting because the partner projects we have actually is spread over quite a broad range of different applications using PharmaShell. We have 2 projects with monoclonal antibodies, and the aim there is to actually to provide a possibility to have highly concentrated monoclonal antibody suspensions, as well as getting longer acting, so slow release monoclonal antibodies. So there are a couple of those. We have a couple of peptide projects together with I say all our collaborations now, it wasn't that way a couple of years ago, but now it is with the very biggest companies there is actually in the world. So that is really promising, I think. And then we have also one project that is extremely important, I should say, but also interesting, and that's a local administration in tumors. And so that is a project that has progressed quite well during the autumn. And we are expecting to see that we see a possibility for a broader collaboration in that field, and that could potentially be the first one going into broader development agreement. But you never know. It might be shifts in decision-making processes in the big pharma is sometimes slow, and you really don't know where they go.
Okay, very nice. Of course, very curious to learn more about that project you just mentioned. And let's put a spotlight on monoclonal antibodies. Monoclonal antibodies is a very mature product, it's a very mature space that is growing very fast. It's growing very strong in the last years, and it is also projected to grow in the decades to come. So monoclonal antibodies is a very interesting space. What are the major limitations you would say that nowadays monoclonal antibody formulations have? And when talking about these major limitations, I would also like to ask you about the different ways how monoclonal antibodies are administered today. I mean, you have IV injections, or you have subcutaneous injections.
Yeah, I think most antibodies today are administered through IV actually, and that takes time. Of course, you need to be in the hospital and have infusion, so to say, for an hour or so, or could take longer than that. There are products that have a sub-Q administration, but normally those products have a very high volume. They need some pre-preparation to be able to administer those volumes subcutaneously. So the need, I think that pinpoints the need today is actually to be able to have higher concentrated suspensions of monoclonal antibodies. What happens if you don't have the shell around the monoclonal antibody is that they sort of entangle when they have a higher concentration, and then they become a gel, and the gel cannot be injected. So that is a problem. When we put a shell around it, there are no sort of arms or so that can entangle, if you want to view it that way. So we can make much higher concentrated suspension. You can say a normal limit for an antibody today might be 150 milligrams per milliliter, maybe a little bit higher. We can easily go up to the double of that and even higher. So that will make a huge difference if you want to have a long-acting monoclonal antibody administered subcutaneously. And having that, there is a big need for that in the market. So we really anticipate that we might proceed with the 2 monoclonal antibody projects that we have, and also get more of those in-house.
That sounds very interesting. And another topic when talking about monoclonal antibodies, some monoclonal antibodies that are administered today, it's like combination preparations with hyaluronidase. Which is like an enzyme when you inject it. I guess you know about it.
Yes.
I mean, just for the audience, is this something that you don't need this helping agent anymore, like with the pharmaceutical technology, so that when you want to inject an antibody at a high concentration?
Yeah, the hyaluronidase, what it does, it creates sort of a cavity where you can inject something subcutaneously. And that is one way of doing it, sort of, but then you can think of the combination of hyaluronidase and suspension using PharmaShell. That could be even you can even prolong the release profile of such a product. But then again, the hyaluronidase is not something that you really would like to use, actually, I would say, because it's sort of, it interacts with the tissue in a way that you really don't want. Okay. But it is a means of injecting high volumes. Yes, it is, and it is used. Yes.
So in summary, there's really a potential for the pharmaceutical technology of improving monoclonal antibodies, the formulations as they are today.
That's the way I see it, and the way you can when you are on these conferences that we attend in Europe and the US mostly, when talking and when showing that we have a possibility to do what I've just said with monoclonals, we have a very high interest from a lot of the big pharma companies.
Okay, that sounds very interesting. And coming to my last question for today, how's your outlook for 2024? What are your milestones? What are your goals for 2024? What you would like to reach?
Absolutely a high focus on the next 22 project. You should expect that we can provide you with data from our first PK study, so that we can show that we have a long release of the GLP-1 product, liraglutide. So that is definitely high focus and high priority for us. You should also, and this is of course something I might not be able to tell you, but we will proceed with the Novo Nordisk project. And like I mentioned earlier, I foresee that we will reach a few of those really important milestones in that project. Whether that will lead to going into a license discussion this year or next year, I can't say today, or if it will be stopped. It all depends on the data, of course.
Absolutely.
But I foresee that we work closely with them. We have already today a wonderful collaboration with them. It's really a great team to work with, and I really see that the preclinical evaluation they want to do prior to sort of a decision how they want to do going forward or not is really going to progress during this year.
Sounds very promising. Your fruitful collaboration, of course, with Novo and your own in-house Next 22 project. And with that, I would like to thank you, David, for coming here today. It was a pleasure to have a fireside chat with you.
Yeah.
So thank you for coming.
Yeah, thank you so much.
Thanks.
Thanks.
Bye.